You have probably heard the term blood bank somewhere, but what image comes to mind? Perhaps you think of a joke about a place where vampires go to make a withdrawal. Maybe you think someone who has needed a blood transfusion, or maybe you have even donated blood yourself. Today, you will learn about blood types and what happened in a blood bank. Blood then pins of life force coursing through thousands of miles of arteries, veins, and capillaries. Those blood vessels carry blood to every other organ, keeping them functioning and thriving. Our red blood cells carry oxygen and nutrients. Our platelets stop bleeding and help to heal our wounds. Our white cells protect us against infection. And the yellow liquid plasma in which they are suspended carries an array of proteins that regulate bleeding and clotting. The two most important factors to consider when giving a blood transfusion are the antibodies and antigen present in the patient and in the donor. An antigen is something that can result in an immune response developed. Hundreds of antigens are found on red blood cells. Let's talk about transfusion medicine testing. Pre-transfusion testing include blood typing, antibody screening and identification, direct antiglobulin test, indirect antiglobulin test, transfusion reaction evaluation, cord blood testing, Kleihauer-Bet kit to detect fetal maternal hemorrhage, and platelet antibody testing. It's the detection, identification, and quantitation of antibodies involved primarily with red blood cells, although white cells and platelets may also be involved.
Right.
Transfusion service work primarily with patient blood. Primary areas of responsibility include blood typing, antibody detection and identification, compatibility testing or the cross-matching, blood component therapy or the chemotherapy, transfusion reaction workout, autoimmune hemolytic anemia workout, hemolytic disease of the newborn works out, determining the Rh immunoglobulin eligibility. Blood banks work primarily with donor blood. Major areas of responsibility include donor recruitment, donor screening, blood collection, testing, typing infectious diseases screening, blood components preparation, component preservation, may provide reference lab services, may store rare donor blood. Therapeutic apheresis refers to a specialized medical procedure in which blood separation technology is used to remove abnormal plasma constituents or blood cells. Whole blood is removed from the patient into a blood cell separator which utilizes centrifugal forces to separate all blood into its individual components: plasma, platelets, white blood cells, and red blood cells. Plasma or a specific blood cells type can be removed and replaced with physiological fluid or cell to treat relative uncommon but usually life-threatening diseases.
Stem cells are collected using a procedure called apheresis, where only the stem cells are separated and collected during donation. The remaining blood components are safely returned to the donor. This is a non-surgical procedure and takes approximately 4 to 6 hours. A bone marrow harvest is relatively simple and typically occurs in the operating room. Bone marrow is aspirated, or sucked, out of the bones by inserting the needle into the bone multiple times. A typical bone marrow harvest takes about 2 hours and involves the removal of 1 liter of bone marrow containing the stem cells. The most common type of cell therapy is blood transfusion, and the transfusion of red blood cells, white blood cells, and platelets from a donor. Stem cell therapy, also known as regenerative medicine, promotes the repair response of diseased, dysfunctional, or injured tissue using stem cells or their derivatives. Stem cells can be administered in a variety of fashions: IV stem cell therapy, intrathecal, side injections into problem areas, knee, hips, hands, etc. Coagulation, also known as clotting, is the process by which blood changes from a liquid to a gel, forming a blood clot. It potentially results in hemostasis, the cessation of blood loss from a damaged vessel, followed by repair. The mechanism of coagulation involves activation, adhesion, and aggregation of platelets, as well as deposition and maturation of fibrin. The prothrombin time, sometimes referred to as PT or pro-time test, is a test to evaluate blood clotting. Prothrombin is a protein. A prothrombin time, PT, test measures how long it takes for a clot to form in a blood sample. An INR, International Normalized Ratio, is a type of calculation based on PT test results.